SCHOice, the best choice for your success. Accelerated, high-performing cell line development at Samsung Biologics. Hi everyone and welcome. My name is John Gill. I'm the Director of Cell Line Development, part of the CDO R&D team at Samsung Biologics, and today we're talking about SCHOice, the accelerated high-performing cell line development at Samsung Biologics. SCHOice, the best choice for your success. The true significance of cell line development in any drug life cycle is something that's often overlooked. The cells act as living factories for biopharmaceutical drugs. It's the very first and critical step of biologic drug development, with many things to consider. The quality and safety of the therapeutics need to be guaranteed during the entire life cycle, starting from CLD. High productivity cell lines play an important critical role in lowering your cost of goods, and selection of a robust host can reduce the run-to-run variability with more consistent drug substance. The main goals of cell line development can be varied, but generally involve regulatory support through documentation and confirmation of monoclonality, and robust clone selection, which play a critical role in the isolation of highly productive and stable cell lines. With this in mind, we introduce SCHOice, Samsung's Super CHO cell, proprietary and internally developed, the best choice to start your therapeutic development journey. The cell line itself is composed of a CHO-K1 cell with a GS knockout selection system. The knockout was developed using Talon technology. The cell line can deliver high productivity with 7+ grams per liter, and it maintains high viability, over 90% through day 21, one of the keys that I'll be talking about throughout the presentation. The advantages of SCHOice. The Samsung platform. First, I'll take you through the development of the host system. The GS knockout cell line development was initiated with the CHO-K1 received from ATCC and adapted to CD-CHO media. From there, we initiated electroporation or transfection of the host cell line with Talon mRNA to knock out the GS gene. We proceeded to the genome cleavage detection assay looking for positive pools, which were carried through the single-cell cloning stage. We went through 3 rounds of screening, 2 passaging with and without glutamine media that were sandwiched around the gene sequencing itself to confirm the true knockout of the targeted locus. Finally, we went through media adaptation, or more to the point, we went through a switch in the base formulation of 16 subclones from the CD-CHO formulation that we initially started with to the Excel CD-CHO fusion media, resulting in 5 final subclones. As I mentioned on the previous slide, the next stage was media adaptation for subclone selection. The process for going through this was passaging the cells at 500,000 cells per mil in the Excel CD-CHO fusion media from Merck. They were passaged every 3 days for a total of 33 days. At the end of the 33 days, we initiated a 6-day batch culture just to look at the growth rate over that time compared to our CHO control that allow us to select for the top clones to move forward for the performance test. As you can see, the doubling time for the individual clones that were selected is quite robust. We're looking at 18 to 21 hour doubling time compared to the 25 hour of the CHO control. And the peak VCD was also higher during that 6-day, with a peak VCD of choice 5 at 8 million cells per mil compared to the 4.2 of the CHO control. Now with the knockout of the individual clones and the selection of the top 5 candidates, we froze a research cell bank and then initiated the SCHOice performance test. Here, we carried through IgG expression in all 5 of those hosts through our full cell line development platform. So that includes transfection and plating 10 96 well plates, 5,000 cells per well, going through the stable mini-pool selection process with static screening by octet. As we moved the top 10% of those pools through the ranking process, we performed a 24-hour QP assessment, followed by an 11-day fed batch, And ultimately, in the shake flask, a 14-day fed batch production study was used to screen the individual mini pools that would be carried through the single-cell cloning stage. It's important to point out at this stage that the P-SBL plasmid map was not optimized. This was one that we'd pulled off the shelf, and we will discuss additional vector technology later in the presentation. As we move through the performance test ranking workflow, I won't spend too much time here, but point out that during the performance test, the first stage of electroporation, we can see that two, host clone 44 and host clone 49, were not transfectable. You can see in the transfection efficiency in the table on the left that the host clone 44 had a transfection efficiency of only 10%. This was only marginally beat by host clone 49 at 14%. So while this was enough information for us to remove those particular candidates from the performance workflow, we did carry out the additional study through the stable pool selection, the 24-hour QP, and the 11-day fed batch to make sure that we had a robust data set allowing us to select the final candidate host cell line. The next stage of the development workflow is the stable pool selection. As the cultures grew in the 96-well plate, going through glutamine removal and selection process, we passaged the cells 4 times, 1 to 4, looking to normalize the confluence and cell growth. From here, we screened 960 stable pools, so all the wells in the 96-well plates, by octet to give us a ranking of the individual wells. What you can see in the result graph here is a significant increase in not only the overall titer in mgs per liter over the choke control, but a bump in the mean expression level as well. So the starting point here is we have good transfecting clones and very good productivity as a readout and the first stage of our performance test. So we took the top 10% of these pools and moved them forward through our process. As we continue our journey through the cell line development performance testing workflow, we are ultimately at the 14-day fed batch screen of the mini pools. Here, the evaluation method is done in shake flask. The top 5 pools from each choke control and the individual clones were carried through a 14-day fed batch in the advanced fed batch media with feeds starting at day 5. The analysis was titer by octet and the cell growth by vi-cell. What you can see in the graph below on the left side is the choke control. Typical expression that we see for an IgG1 through our workflow is a little under 3 gram per liter, so 2.9 gram per liter at day 14. In comparison, the SCHOice with the top individual host clone, total 56, you can see that we're able to hit 5.7 grams per liter, so almost a 200-fold increase in the individual titer at the mini pool stage. This allows you to move through your process development by transferring mini pools through the upstream process development to generate material for formulation development, analytical development, and your downstream development. From here, we took the top expressing mini pools and went through the single cell clone isolation stage. One more piece of data to show and share as we go through the performance testing and get to the single cell cloning is the performance of the cell line as a host and the potential benefits of moving forward with the SCHOice cell line. What you can see here on both the top and the bottom is a robust viability. The choke control is something you would see typically at the end of a stable pool 14-day fed batch with viabilities ranging from mid-40s to the high 70s. You compare that to the SCHOice cell line where the bottom of the individual pools that were screened is in the 70%, and we had even mini pool screening at the day 14 stage at over 90% viability. What this ends up yielding is a robust viable cell density profile over the course of the 14 days. When compared to the VCD of the choke control, which you can see peak around day 7 and continue to drop through day 14, the VCD will match that peak VCD around day 7 and maintain over the course of the remainder of the study. What this allows us to do is increase the overall process yield, but also result in higher product quality because of the lack of low viability. As we move through the performance test, we're ultimately jumping to the final 14-day fed batch process, screening the final isolated single-cell clones. The process here, the media, was an XL advanced fed batch formulation from Merck with a feed of Cell Boost 7A, 7B from HiClone, and a working volume of 50 mils. The graphs on the right compare the stable pools and the resulting clones from both the choke control and SCHOice that was shown on the previous slide with titers of 2.9 and 5.7, and the resulting clones that were isolated from those stable pools. With the choke control, you can see that we were still able to get a sizable bump from 2.9 to 3.4 gram per liter, but in the SCHOice host cell line, we went from that 5.7 gram per liter, which was already quite high, to isolation of clones that were producing 7.6 gram per liter. So as we demonstrate the ability of this host cell line to generate a clone with sufficient titer and expression levels of over 7 gram per liter, it allowed our upstream development colleagues to work on the high manufacturer performance evaluation. Here we're showing the result of a seeding density and feed optimization study, where we took the clone that was producing 7 gram per liter on the previous slide and went through a full 21-day fed batch process. What we're trying to demonstrate here is the robustness of the cell line. In the middle graph of viability, you can see that even at the tail end, at 21 days, the viability of this individual host at multiple different seeding densities is still above 90%. And as pointed earlier in the presentation, what this viability does is allows us to hit peak VCD as early as day 4 and day 5, depending on your seeding density, and that's maintained through the course of the entire study. Now, while from a cost of goods perspective, you may not want to run a full 21-day fed batch manufacturing process, it does point to the robustness of the cell line not being at the edge of failure. In the eventual result, from a titer perspective, just from this particular study, not only was that 90% viability maintained through the 21 days, but the titer reached or achieved at day 21 was over 11 grams per liter. As mentioned earlier, we had not performed any of the vector development prior to starting the performance test. Here, we're demonstrating the additional Samsung platform improvements via vector design. The initial design for the vector that was used during the performance test was the PSBL1. It's a mouse CMV promoter, followed by the human CMV and human EF1 alpha introns. The design testing itself was looking at 4 different promoter sequences in combination with the introns that were used for the previous experiment. Here, we looked at the mouse CMV compared to rat, the human EF1 alpha, and the SV40 enhancer plus human EF1 alpha combination for PSBL9A. The gene of interest that was used for this particular vector design study was GFP, and what you can see in the table or graph on the right is the fold increase as we look at the additional vectors that went through the study. Everything normalized to the PSBL1 GFP. You can see that the plasmids far to the right, PSBL8A and PSBL9A, saw close to a 200% increase in GFP expression. So with the exciting 200-fold increase in the GFP expression, we took the PSBL9A base vector, and in addition to the IgG1 that was used for the performance test earlier, we looked at FC fusion and bispecific expression. As mentioned on the previous slide, we took additional protein targets through the Samsung Vector Performance Evaluation. Here, again, on the left, we're showing the 2-fold increase in GFP expression from the PSBL1 to the PSBL9A. And that increase as a percentage was shown with the expression of both molecules, the IgG1 and FC fusion, showing a consistency of expression regardless of the gene of interest. Faster and better, together with SCHOice. Throughout the talk, I tried to emphasize the importance and impact of selecting the correct host cell line. Here, we're talking about fast development timeline from gene to drug product in a little over 10.5 months. Kicking off with the cell line development activities from transfection to single cell isolation and cryopreservation of your research cell bank, it triggers additional process development activities from your cell line stability study, your MCB manufacturing and characterization, your upstream, downstream formulation and analytical teams, and generation of your tox manufacturing, Tox manufacturing itself can initiate within a 5-month, 6-month timeline, ultimately allowing us to move through a cGMP drug substance manufacturing process between months 8 and 9, progressing to gene to drug product in 10.5-month timeline. With further support from regulatory documentation, it truly allows you to accelerate to your IND. Get to IND faster and better with SCHOice. The key advantages of the SCHOice, the best choice for your success, the Super Cho cell line, delivers high productivity with over 7 gram per liter. It maintains a high cell viability, over 90% through day 21, leading to a more robust and consistent drug substance and drug product. And it's a key part of Samsung's Faster and Better service, your success to IND and BLA. The fast timeline of cell line development for the first primary screening, from transfection to single-cell isolation, 6 weeks, with additional 7 weeks for the secondary screening, 14-day fed batch processing, and the cryopreservation of your top 5 RCBs. This is a critical part of the seamless one-stop service, fully integrated development and manufacturing here at Samsung Biologics, with excellent quality and outstanding capability, with over 59 global regulatory approvals. The cell line itself has competitive pricing, no binding structure, or no royalty payments. And truly our goal is maximized client satisfaction, both from a cell line and scientific perspective, but also with dedicated PMs to ensure successful project execution. As your most qualified, world-leading CDO, CMO, CRO partner, Samsung Biologics delivers your success to IND and BLA faster and better with maximum flexibility. We promise the best. SCHOice, the best choice for your success.